On the other side is the School of Engineering. We're going to go out into the courtyard up here called the Court of the Sciences, because it's the central courtyard around which most of the sciences are circled, except for a few oddballs like physics up over here, which is north of the dividing line, if you will. Okay, more like 50 instead of 35. All right. So this is the Court of the Sciences. So if you're a science major, this is where you spend much of your time. This building here is mathematics. The School of Engineering. Looks like it's a small building. You got to go and look on the other side, building after building down there. So the School of Engineering. Over here is psychology. Earth and Space Sciences here. The brick building down there where we're going is chemistry and biochemistry. The concrete one down there is molecular biology. If you look through the trees down there, the brick building, that's life sciences. And the tall one on the far side is the Cancer Center and the School of Medicine, School of Public Health, School of Nursing, are all down over there. Slowly migrating this way as the medical center takes over the campus. This is a campus of 35,000 students. 500 of those students are in the School of Medicine. 40% of the faculty are associated with the School of Medicine. We estimate that of the entering science majors at UCLA, 95% aspire to go to medical school. So you will be in with a large number of your compatriots if that is your aspiration. So here is where most of the classes take place. You see the portico that we're going to go through, past over here. Inside that portico are the large lecture halls where the chemistry and biochemistry classes are taught. It's going to be typical of your classes here. Those rooms range in size from 235 to 350. I'm now teaching the smallest class I have taught in 25 years here at the undergraduate level. I have 30 students. I'm amazed. I have never seen this before. I don't know what to do with such a small class. I guess I scared them all away. So we're going to go over to the steps down here by the chemistry and biochemistry building. We'll then split into two groups. We don't have any planned schedule. Gloria and I will have to split you up, but we're going to walk through. We're going to see the following. We're going to see some instructional labs. The instructional labs are where the courses in life science or chemistry and biochemistry are taught. So that might be microbiology, molecular cell and developmental biology, or biochemistry. We'll also look at a research lab. The research lab is kindly we're being able to go in there because a new faculty member has just arrived. It's being set up, so it's not fully equipped. We'll also, if we keep the group, can get the group small enough, we'll look at some of the research facilities. When you go through the building, what you'll notice is that the vast majority of the building is not devoted to undergraduate instruction. It's devoted to research. So that most of the building that we will go in, the main floor, there are 5 floors in the building. The first floor and the 6th floor are devoted to teaching, and the 6th floor is only half the building length. The rest is devoted to research, so that each faculty member will have a large research lab, and we'll see an example of that as we go through the building. Are there any questions? No? We're all set. Have a question? Okay. How am I doing for time? Gloria's expecting to call.
You're about 115.
Should be there.
Chilly out here, so I saw you.
We have about
Pressure on
50,
here.
so it's going
Oh,
to
this is not
be very difficult with this many. They didn't keep the number down like they said they would. So we'll have to split it in two. Which order do you want to go? I was going to walk through the south wing and then fourth floor. May have to break each group into two to go in.
Then I'll start at the top.
Okay, so we're here and this is the chemistry and biochemistry building. We're going to split in two. Just to point out, down here is the bomb shelter, which is one of the myriad of eating establishments here at UCLA. So we're going to split in two. This is Gloria Turner. Gloria is a professional researcher here at UCLA. She will be leading about half of you through, and she'll start going up, and we're going to go down the other half. So we'll split, and the first half, do you want to take your half
Yeah,
in?
you're going to be, I don't know if Dr. Weiss told you, we're going to take you through upper division teaching labs to get an idea of what kinds of equipment and facilities that we have for our upper division biochemistry, and sometimes biology students take this course, as well as his own research lab. I work with Dr. Weiss, so we'll tour those labs. And then we have a new faculty member, Dr. Carla Kohler, and she's just setting up her lab. So I thought it'd be fun to see what it's like to be setting up your own research lab someday. And then we have an instrumentation facility that this might be a little awkward going in
I
there.
would take and split the group.
Yeah,
into two
because this
and
is
go in
a
very
facility that's
briefly.
heavily used, so there'll be people in there doing things, and there are also radioactive things going on there, so we have to be very, very careful when we go in there, but just so you can get an idea of what instruments we have, and then through the computer lab facilities, so.
Okay, so the first half, I'm going to arbitrarily sort of divide the line right here.
Yeah, Cindy's there, yeah.
So this,
Yeah, okay, thanks, Sean, have fun.
okay, this group will go with Gloria, and this group will go with me, and you're off.
Okay, all right, so. Let's go. So we'll try and, since we have such a large group, we'll take the stairs, okay? You guys will have to get some exercise. So this is our reception area here for chemistry and biochemistry. Hi, Ron. So if anyone calls a department, someone in this office will answer and try and direct your call to the right person.
They can't, they'll just try.
Huh?
They can't, they'll just try.
Yeah, exactly. And some are better than others at that. Yeah, so, and then we have a research stock room. So it's really nice. Sometimes paper supplies and glassware we can purchase there. Normally we have these kinds of things, but if you need something that you're maybe doing a new experiment and you need something that you've never used before, you can run down here and usually find it. Yeah, well, yeah, right, no, you have, you have account numbers, and those account numbers correlate to a grant, a research grant. So they just kind of, it's sort of like having a checking account, and you just write a check, only we just sign an account number. Yeah. Oh, Betty! Yeah, well, this is just half of them. Do you have a second? This is Betty Yeager. She coordinates the upper division biochemistry lab for our department, among a myriad of other things.
5, 6, and 8.
She's going to...
They've got to be 20 or over.
Yeah, so we're actually, the first thing we're going to see are the labs that she's in charge
You'll
of.
have to see Charlie and Dave.
Okay, yeah, so we have 2 really senior graduate students who are taking the course. And actually, Dr. Weiss is teaching that course this quarter, yeah.
So you're from you're from Mount Sac
Oh,
Valley.
yeah.
All community colleges all in this.
Yeah, this is a TAP transfer, right?
What's TAP?
The honors program.
Transfer lines.
Yeah. Okay, so we're gonna have to have Betty's into detail and practicality. Well, all the labs have what they call safety bars on their shelves, and if you have large cylinders or gas tanks, they have to be secured. So we have a number of, our acids are kept in special cabinets, and so we do this really for that reason. If there is an earthquake, most of those hazardous things will be contained.
And also I see basic
Oh,
gas.
yeah, we have. And we also take, I've been trained in like emergency response training by the fire department, and they always have somebody in each lab that, is familiar with what you do in case of a disaster.
And like right now you're not there?
Neither is, the only person there is our lab helper. And yeah, that's bad. Normally there's a few more people, but everyone went to lunch. So these are the biochemical division labs. So we're gonna, actually, you can see the students in there. It's Charlie Boyd, he's TAing the course. Hi, Charlie. We're gonna go. Well, is that a good idea? Betty said we could, but
Yeah, it's not too crowded.
Okay, these are all junior college students who will be transferring to UCLA. So they're just taking a tour of our facility.
Okay.
So this is Charlie Boyd and Dave Mole. They're 2 senior graduate students who are TAing this course. So they're actually helping the students with their experiments. So we all. Yeah, and these are UCLA students. Yeah. Trying to figure out what they're trying to figure out.
Well, they're trying to figure out how platelets are activated.
Oh, how platelets are active. Oh,
Right. So this is we're doing today is a lipid flip-flop experiment where you watch phospholipids that are normally on the inside of the cell membrane flip to the outside, and then those in turn activate an enzyme which causes the platelets to aggregate.
That's kind of a neat experiment.
So this is our nice equipment and stuff, and we are doing some other things like isolating lipids from the platelets, isolating lipids from the platelets, and then we're going to run TLC plates like this and separate the lipids and identify which ones are labeled.
Well, that's nice. That's an experiment.
Yeah. It's really a detailed experiment. Correct. So.
Do you have any questions that you would? No, just.
Yeah.
Okay, well, I'll take you into there. There's another lab that's not in. There's really nobody in there, right? So we can.
Yeah.
Next thing. So you can. These are all of our spectrophotometers have computers now. I know. So in this room are all the centrifuges and our liquid scintillation counters. You can see how many, and these are supplies. That's a really well equipped lab. Betty's done a wonderful job. Yeah. So you guys have it. Well, we have little tiny actually, I don't see any little tiny centrifuges, but like a clinical centrifuge or a microfuge, they have those as well. So you can kind of wander around here. What Betty does actually she does it. How this lab has always been run is that divide you into groups. So usually 3 to 4 students work together on these projects, because the projects are quite involved and require making solutions and doing centrifugations and labeling with radioactive isotopes. So there's like usually 3 or 4 parts to every project, and you do 3 projects in this lab. So it's quite an intense and really you learn a lot. You learn an awful lot. So she takes a picture of each group and puts it on the bench. so like sometimes when you come in, you'll see all these groups of students. She really does a beautiful job, or everything is so organized, right? When you see some of the other research labs, you can appreciate how organized she is. But most of all the labs in chemistry and biochemistry have fume hoods. So because even though we do biological experiments, a lot of times we use volatile chemicals like phenol, a very strong acid, or HCl, something like that. So we do all that kind of thing in the hood. Plus we also work with things that this is better to work with them in the hood. So almost every lab in this building has a couple of hoods in it. So do you have any questions about the teaching lab? Or they have this course. There's a total of I think 6 experiments. So they rotate the experiments, and I think right now there's 3 that they're kind of doing all the time because they are the most informative experiments. Some of the older experiments, the methodologies have changed, the techniques have changed, and they want to teach their undergrads the latest in everything. So yeah, that's kind of nice. So okay, we're gonna go downstairs. This is just one flight, and then we'll go through Dr. Weiss's research labs. Okay, and I'll check and make sure Cindy hasn't Cindy Chan. She's a lab helper and also a UCLA sophomore, right? So, oh here, I'm sorry. One second, excuse me. Just come on in, look around. Yeah. Oh, yeah, you did. I did. What did you need? I didn't know you could look in the figure out here. Yeah. Okay, she's Okay, all right, sure, excuse me. Okay, so this is kind of what most biochemistry labs look like, I think, because we all have these refrigerator boxes because we have to do things in the cold. We have a lot of freezers and refrigerators since we have to keep a lot of solutions and strains in the cold. We have pH meters, chemicals. We have, these are the little centrifuges. I don't know if some of those little microfuges. We try to do experiments on a micro level, right? It's just so much nicer. You use less material, and especially when you're doing pilot experiments, you try to do everything small, and then you can scale it up. So, and most molecular biology is done small. You do everything in little tiny bullets. So I don't know if you've isolated DNA yet in your courses. So probably some of you have, and some of you haven't. So you probably did those all in little tubes, or yeah. So those are we call them bullets or microfuge tubes. Yeah, and. So yeah, some people have more colorful benches than others. Kate has these flowers on her bench. So these, the graduate students, this is kind of their home, right? Because they're here working a lot, so they like to be comfortable. And all labs now have to have 2 or 3 computers, and I guess you're all being taught to use computers. It's just so integral now in our lives. It's just amazing how important it is to use computers. And labs, we just got a couple of new iMacs. We're all Mac people here. So I don't know if you're Macintosh or IBM.
Doesn't matter.
Doesn't matter. Yeah, well, now that they're sort of the same, I guess, really. But back in the old days, IBM, you had really DOS and do all this.
Right.
I mean, DOS, and it just was so difficult to work with IBM computers. Once you've worked with a Mac, kind of a menu-driven program. But now they're all menu-driven and mouse-controlled. Yeah. So I'll take you over to the little lab. How you doing, Cindy? Okay. She's making a solution for the first time. Very good. So this is, I'll let everyone come on in here. So we work with a fungus, Neurospora crassa. So it's really a model organism because it's a eukaryotic organism, so it's like us. It has a nucleus and mitochondria and all kinds of vesicles, and also it has different developmental pathways. So it's even though it's a simple organism. I'll show you what it looks like. I've got a bunch of plants over here. Yeah, because there's a genetics course on campus, LS4, and they're doing interactive CD-ROMs for the course. So they are going to actually do a module on Neurospora. So the wild type Neurospora is this orange little fluffy stuff. See, it's this little fungus. So it grows by making what we call a mycelium, and then makes aerial hyphae, and on top of the hyphae are little balls, which are conidiaphores. So there are like millions of little spores in this tube, so we have to be careful not to open it up. And then we have a mutant that's albino. So this mutant is defective in its carotenoid biosynthetic pathway. And so just within the last 10 years, most of the genes that are in this pathway have been cloned, and so we understand why this is white. But it comes in really nice when you want to mark, like say you want to do a genetic cross, but you can't really follow that cross because it doesn't really, or that particular marker, it doesn't really have an effect. But if it's close to one of these albino genes, then you can just follow your marker with this because it'll always be white. And that's exactly what we do with one of our arginine biosynthetic genes. It's since we work with a number of them, we want to know which is which one, we mark the one with the albino. So this is like velvet, so it's like a really strange guy. See, it's just, it got stopped very early in something, and it just kind of does the same thing over and over. You guys might appreciate this. I went to a plant seminar once, and someone was talking about these mutations in this particular plant, how it's very early in development, and so the organism just keeps doing the same thing over and over, and that's how we get broccoli and cauliflower. It just, this little thing that just keeps going and going is that kind of a block in the plant. And so this is kind of the same thing for this mutant. It just doesn't get started. And then we have peach. We have different colors. We don't know really what this mutation is. And these, like, really strange guys are, their mutation is in adenylate cyclase. So which is a very important enzyme because it controls cyclic AMP. In your, so I mean, see, this little guy does exactly what we do. Yeah. So it's kind of, but it's much easier to work with. We can just, grow this up, grow large amounts of it, throw it in a bead beater, make an extract and assay, and make DNA, make RNA, which we can't do with people, right? We can't, not yet anyway. So this is, we work on arginine metabolism. So we use Neurospora as a model organism. And on this floor, almost everyone does the same sort of thing. They have, they're asking really interesting biological questions, but they're using a simple organism. So across the hall, actually the two fellows that were teaching upstairs, they work across the hall. They're working on Colobacter, which is a bacterium. So it's a prokaryotic. Neurospora is a eukaryotic. And then across the hall from them, someone's working on Saccharomyces cerevisiae, that's baker's yeast. So it's a eukaryotic organism, but it's very simple. It's kind of like bacteria. It just buds and divides and buds and divides. So neurospora is a little more interesting, I think, developmentally. And then someone's working on Chlamydomonas reinhardti, which is an alga. And then we have Drosophila on the floor. So we have like a lot of Drosophila melanogaster, which is like a little fruit fly. And a lot about development is being learned from doing basic research in flies. So it's kind of, you know, all the things that I think all the research that goes on really contributes to understanding how things work in human beings. And I'll tell you, we really are so far from understanding most things, you know, really. There's so many questions to be answered. Oh, so many. So when you sit in your classes, you think, oh yeah, they know everything. We don't know anything. And so when you have a good teacher, they'll tell you that. They'll let you know how little we really know, and how things are just in 10 years, things have just changed so much. So we'll go now. Okay, so this next facility is our instrumentation facility, and we'll peek in and see how busy it is. But in there are like hundreds of thousands of dollars worth. So Rachel, these are junior college students. They're going to transfer hopefully to UCLA next fall. So they're taking a tour of the facilities. Okay. Yeah. They met Dane and Charlie upstairs. Okay. So we can get everybody. Yeah, just kind of. These are behind you are liquid scintillation counters, and we use those to determine the amount of radioactivity we have that we're using in our experiments. They're not radioactive, don't worry, but they have like a photo multiplier tube inside, and when you want to like determine the amount of radioactivity you have in a certain sample, you dilute it up and mix it with a cocktail, which is actually a scintillation fluor, which enhances the photon, like leaving the radioactivity, and you can count that. And so that we have standards, so we know like what so many CPMs are equivalent to, and so that's how you can determine how many CPMs you have in your sample. So that's behind you, and then in front of you is this is an alpha imager camera. So if you run I don't know if any of you run any DNA gels. Have you agarose gels? Okay, so you stain them with ethidium bromide, and then let's see. So yeah, well, this has got. You can also look at protein gels on here. But there's a transilluminator, and then you can. Too bad no one's here with a gel because we could see it. So you put it on here, and then close this up, and then turn the computer on, and the image of the gel is on the computer. So you can and you take a picture with a camera, and the printout comes out of the little computer. So it's really nice. So we used to have to use, we had handheld cameras that used Polaroid film, which costs about $2 a sheet. So now this is about 7 to 10 cents a sheet to take a picture of your gel, just to document your experiment. So this is kind of, it's too bad no one's got a gel. So then right here is a phosphorimager. So this has changed how we do a lot of things. So if you are doing an experiment that, say you are labeling up, like I do a lot of northerns or southerns, so you run some RNA or DNA on a gel, and then you want to see how much of your specific gene that you're working on, like want to maybe do a restriction pattern, or if it's with RNA, you want to look at its expression, then you do what we call a northern. Do you know what northerns are? Okay, well they don't have to be done with radioactivity, but they often are. And so you actually end up with a membrane that has radioactivity on that. And so you expose that membrane to a phosphor screen, and that screen then can be scanned in this phosphorimager, and then you can quantitate the amount of radioactivity that's on your membrane. So, and this is also done just very similar to the alpha imager camera. You scan and the image is digitalized, and then you can look at it on the computer. And then we have programs that allow you to quantitate that and manipulate it as well. Okay, does that sound?
Close enough. Technically, what happens is the radioactivity hits this really expensive rare earth ceramic, takes electrons up in energy where they get trapped. Then you run a laser on it, and the laser lets the electrons drop, and when they drop, they glow. So what it does is it takes the little screen and it scans a laser past it, and then it glows back at it and reads the glow.
So it should
that's
be the image.
proportional
Yeah. So were you doing the oligosynthesizer or?
There was a bum column, so I was fixing that.
Oh.
Anybody want to see what it looks like?
Yeah, this is where we make little short pieces of DNA and sometimes long pieces of DNA. So it's a DNA synthesizer.
Basically, it makes.
And this is Martin Phillips. He's the director of this facility.
Okay, basically what you have here is actually a little bit of glass with the base T on it. You then squirt through chemicals to make either T, G, C, or A, covalently linked to it. Okay, you squirt through a whole series of reactions, all of these different chemicals. It's basically just an automated way of doing. You could do this, by hand, right? You have to be injecting each of these and waiting around and doing it for every base. So it does it instead. Okay, it runs about a base every 4 minutes, and you usually make things on the order of 30 bases long. So you're not making, since your genome is what, 6 billion? We're not synthesizing you just yet, but we make these little pieces that are handy for different experiments. Okay, right now it basically just pumps chemicals through here, running the reactions inside something like this. Okay. And Gloria, that was one.
Yeah, no, that was great. So I don't know if they've seen the spectrophotometers up in.
Okay, well that's another one. It's almost a duplicate of what Betty has.
Yeah.
Some of the other things are somewhat more complicated. They work in different ways. Like that is a device basically for mixing things in, say, a millisecond, a thousandth of a second, and then watching the absorption change. So you watch reactions occur in the millisecond time range, if you're interested in how the rates of reactions. That's a gizmo that lets you look at protein's secondary structure. Those two are calorimeters, which measure the heats of reaction and protein denaturation. So you have this mix of some things are reasonably sophisticated and some are not quite so sophisticated. You can learn how to use this in 15 minutes. That's about 6 hours. That's about 10 minutes. It varies, but it's all things that are sort of generally used.
And the Viacore.
Oh yeah, you can learn how to use that in a few days.
How long does it take to learn to use a Viacore?
Doesn't take long to learn how to use this.
Getting it to do what you want it to do, yeah. It's a way of looking at how I think most people do two proteins, some do DNA and protein, looking at if they interact or don't interact, and if they do interact, the kinetics of that interaction. So you have something fixed to a surface. What are those? Are they called sensor chips, or the surface that they do is fixed? You actually have something that can't move, and then you flow through a solution that actually has what you're interested in checking or learning the kinetics. Professors just joined the faculty, and they're setting up their labs. This is Dr. Kohler's lab, and Dr. Champrose is across the hall. So you can see that she's got some nice colorful cabinetry here, huh? And what's this? Oh. Yeah, so remember I was talking about, come on in, just come on in. You can come around the other side. Just don't, I guess there's really nothing out there for them to worry about touching, right, Carla?
No.
Yeah, okay. Yeah. So both of our new assistant professors use the Baker's yeast, Saccharomyces cerevisiae. Remember I was telling you about model organisms and how how many things that we can learn using a model organism. So both of them use yeast. So they're uncrating their new microscope. Oh, that's gorgeous. So you have a video camera?
Yeah. Huh?
They're gonna call me
Okay.
probably tomorrow.
Okay.
Okay.
Yeah.
Okay. Hi, Christina.
It's the right way. It's this way. It's this way, you know. It's usually
So have you all used microscopes before? Yeah, yeah. So this is a really nice one. Gotta figure out how it goes. See? Everything's always so again, you can see they're just getting things set up. So they have cold boxes and refrigerators. Hi, this is Christina. She works with Dr. Kohler. She's helping her set up the lab. Are you getting tired?
yeah. Ordering.
You guys should do experiments.
Yeah.
Okay. So everything is brand new in here. It's really nice, huh? Oh wow! Look at this Eppendorf. These are the new kind of really nice new microcentrifuges. So this is fun, huh? Yeah, these are gloves for the autoclave, because we have to sterilize a lot of things, so we need to. Well, I didn't show you the autoclaves upstairs. Have you seen autoclaves before? Have you had done?
Neuroscience is your major, then that's the building, your main building. I love this building because this is the chemistry building. I love that building. We got one of our big lecture halls, which is CS50, right in the center. It holds around like 300 students, I think. So classes can get sometimes big, but then they do break it down in sections, so I wouldn't worry about it. Basically, lectures they just tell you the stuff, and you go to discussion when you have more questions, and then you guys answer problems and stuff. Office hour. It's important to go to office hour, you guys. I think you know that already. When you guys need help homework, important to go talk to the professor or the TA. Anyways, TAs are usually grad students, like trying to get their PhD or master's or something. But sometimes, yeah, I mean, they really know their stuff sometimes though. So sometimes, like I had a TA. Well, I don't know. It's my own experience. So I had a TA who didn't know what they were doing. But I wouldn't want to quote you guys on. I mean, tell you guys that. Anyways. Okay, the psych building, well, this whole thing is going to be the psych building. You guys have any questions? I'm just like rushing through everything right now. Because it's just an hour and you guys want to see everything you told me. Murphy Hall is down there. It's like on...
What is it? Why did he bring it up? That bomb thing. That's
what's that? Oh, the bomb threat? Is that it? Yeah. Is that what you're talking about?
Well, what's it?
Oh, it's the administration building. It's
The counseling and stuff?
it's the chancellor's office. Dean. I don't know. I don't know what the deal is, but yeah, I was there when they had bomb threat. I'm like, everybody get out. I'm like, okay. Anyways, you guys have any questions at all? No?
Where are we?
We're still in South Campus. Right now we're like towards in the middle of campus now. Like I said, that was the education career center. Good place. I got a job during the summer there.
Are we
Okay, bye. I had an internship with a company down at Culver City, so good place to go. They hook you up with. I'm sorry, they give you basically they tell you all the opportunities you could get with your major or what you're interested in and stuff like that. We do have like what we call job track, where it's only for UCLA students, and it's like they have a little job listing only for UCLA students. So it's pretty cool. I mean, you get pretty selective in your job decisions after college, or I mean, if you're like interning for something. Anyways, okay. Psych building. All you psych majors out there. Cool? Yes. You have a question? No. Psych major? Cool. Yeah, I mean, I don't know. I always make up story about this. Like, you know how there's the super ego, and id, or how does it go? Yeah. That's the big, the small, and the small. Like, this is for the PhD students, the grad students, and the undergrad students. I don't know. I just made that up, but I thought that was pretty cool. Like, oh yeah. Anyway, okay. So that's psych building. Physics building. I love that building too. Physics. You take all your physics classes. I happen to take like 6 physics classes, and I'm not taking any more. So I'm done. Anyways, okay. Inverted fountain. Who places me? This place. Just close your eyes and you feel like you're running spring. Do you have any questions? You know what? I think when time graduation comes, all the seniors jump in there. I think. I've seen it happen, but I don't think it's a yearly thing. I don't know. I'm not sure. Anyways, we'll try to go over. You guys want me to talk about admission and stuff like that? No. Oh, you guys heard that already?
Yes.
Like admission, blah blah blah, how to get admitted, application process. You guys want to hear that boring stuff? You sure? I thought you heard most of it all day. You've been hearing it all day. So is it okay with you if we don't go over?
Any jokes?
History of the campus? History of the campus, okay. UCLA was established in 1919, back in downtown, then moved here in 1921. First 4 buildings are going to come up pretty soon. What else? Well, what else can I say about campus? It's 419 acres big, so it's a city within itself. 50,000 people come in and out of UCLA, comprised of 30,000 undergrads, 12,000 grads, a couple of staff, and other people. Anyway, okay. Next. Okay. One of my favorite buildings too is Schoenberg Hall. That's the music building for all of you music majors there, or performance majors. I'm proud of it. I was in the band. I marched the Rose Bowl. What else? I went to football games.
Yeah, I did.
It's cool. What else? That's it. We have the music library there. We also have like performances. Like there's an upcoming performance by I think Wynton Marsalis. If you guys don't know jazz, it's like one of the famous jazz people, whatever. But we do hold like special events there, so it's really cool. Wait, I have some questions. Oh yeah. If you guys want me to talk about admission, now's the time, because we're approaching Murphy Hall. That's Murphy Hall over there. So do you guys want me to talk about admissions?
No.
No. Okay. Sorry. Majority rules. Okay. That's in the department. That's basically where the Slavic languages, our English writing programs are over there. And why does it always come up? The Lesbian Gay Center is also there. It always comes up in my head. I'm sorry?
What is that? The English department?
the English writing program, not the English like writing composition. What else is there? That's it, basically. Asian American Studies are also there. Alright. This is Perloff Plaza. You get tired? No, I'm excited. I mean, I didn't do it. So I'm like trying to get rushed. Low things right now. Alright. What? I don't look that excited. Alright, now we're in North Campus now. We're officially in North Campus. These are the first 4 buildings of UCLA. That's Kinsey, that's Haynes, that's Powell, and that's Royce Hall. They're originally built there in 1921, so really old buildings. Building 1. I told you guys about this building. That building's under repair because of the earthquake, I think. Yeah. Okay. Like the previous earthquake, not the previous one, but the one before that. Yeah. So they're retrofitting it. That used to be where the anthro department. Now they moved it around. Anthro, Chicano studies. I think Asian American Studies was there before they moved here. A couple other departments are there. Yeah, I think that's it. Okay, that's our library. That's one of our 13 libraries here on campus. That's a college library. That's basically where the undergrad library is. We have other libraries such as the engineering library, the music library like we saw over there. The English library is going to be there. What else? Chemistry library. All sorts of libraries. 7 million books total in library system. So, big. A lot. Computers are also here. We have labs here. As a UCLA student, you're entitled to an email account you could use like to log in any computer on campus. Pretty good, pretty fast connection. If you guys know, UCLA was one of the first the first nodes of the internet. If you guys know what that means, it's like one of the first sites, I guess, for the internet. Yes, okay. Next. Well, how are we doing in time? Good? Yeah. Alright. That's the library, okay. You guys could visit there. It's really nice in there. Really quiet, really good place to study. Book stacks downstairs. If you guys get a chance to visit, please visit it. I mean, it's like really pretty. Okay, Royce Hall, our most well-known building. It's Royce Hall. Just because of the imperfections in the sides. See the sides imperfections. Like the two sides are not symmetric, and they're really intrigued by that. I don't want to tell you guys. It's not symmetrical because I think it's modeled after a chapel. So chapel thought, well, you know, God's only perfect being. So it's got
Why are you
imperfection.
hesitant about saying that?
I don't know. It's like you gotta be.
I like the note. You have to say yes. Saying yes.
Yeah. I just thought.
No, we're paying attention.
Alright, that's Perloff Hall. That's architecture department. Design. Classes sometimes are taken there. Sometimes. What else? Okay, I'll go over there later. Okay, coming out, like, if you guys have any other questions, I'm like, just telling you guys what the buildings are, what they have in there. And I feel really dumb for telling that, but I know you guys are really smart. English department is going to be there.
Oh,
It's going to come on.
Okay, so we are going to take
That's, by the way, over there is Dodd Hall. That's basically where the poli sci poli-sci classes are. Behind that is the law school. So if you guys are thinking about grad school, law school, around that nature, we have that over there.
That one
As you
right
guys-
there is poli-sci?
Behind it.
Oh,
Behind it.
What's that one right there?
That's the dot building. That's where poli-sci classes
Well,
are.
poli-sci is
Yeah.
right there. Okay.
So. I'm sorry? Art. This is one of the art schools, School of Architecture. The other design school is Dixon. We're gonna come up pretty soon over there.
Do we test communication?
Sorry. Yeah, we have communications. It's at. Ooh, where is it at? I'm blacking out. Oh, I know. Where is that? Public policy. Half an hour. Okay. Okay. You guys want to stop and look at how close?
Tree barking. Like regular tree barking. Okay.
What else you guys want to know? Story of the campus. Not much. I mean, it's long tradition. UCLA has been here for 80 years, so a lot of people came and gone. I'm like blanking out right now. I know I have a lot of things to say, but I think like most of the admission stuff they went over already, right? You need an application. All the transfer stuff they went over. Like you guys had the whole day over that. Basically, I just want to show you guys my campus. Alright, those are the dorms. There's two types of dorms. One's the res halls, which is the buildings over there. They're like little hotel rooms, but not really. They're kind of small, but not really. And over there is the resident suites, Sunset, and behind it is Saxon and Saxon Suite. Basically, in the suites, you share a bathroom with 3 other people, while in the res halls, you share a bathroom with the whole floor. So they have stalls though, don't worry. They have stalls. I was scared myself. I was like, oh my gosh, I gotta take a shower with other people. It's not cool. But they have stalls, and you know it's cool. I mean, you get to talk to people while they take their showers. It's co-ed. All the dorms are co-ed. I think there is a all-girls floor though in Sproul. Don't ask me why I know that. It's just there. What else you guys need to know? Housing cost, you're around 6,000 I think now, or 5,000 if you live in the dorms. But I think if you live in an apartment, it'll be a little cheaper. Although not for me, because I always eat out at the time.